Good morning and welcome. It is an extreme pleasure for me to be able to talk to you today about one of our latest research. And I would like to start by expressing a huge thanks to Knut and Lloyd for organizing this conference and for making it possible that I can reach out to you today, even though not being physically present in San Diego. The bond that I would like to talk to you about today is the disulfide bond. And I think many of us know this bond is extremely important in many of the recombinant proteins that we produce. Disulfide bonds are formed through the action of protein disulfide isomerase, an enzyme present in the endoplasmic reticulum of all eukaryotes. And PDI can have two activities. On the one hand, de novo disulfide bond formation through its action as oxidase, and on the other hand, reshuffling of incorrect disulfide bonds through its isomerase activity. To act as an oxidase, PDI needs to be reoxidized through the action of ER1, ER oxidase. And finally, the electrons are transferred to molecular oxygen as terminal electron acceptor, thereby producing hydrogen peroxide. Many of us have already overexpressed either PDI and or ERO and shown that this improves protein production. However, not much else is known about the oxidative folding machinery in Comagatella or Pichia. What is known from other organisms is that the activity of ER1 is regulated, and it can switch between an active and an inactive form through the formation of an intermolecular disulfide bond in order to prevent hyperoxidation of the ER. The mechanism has been elucidated both for Saccharomyces cerevisiae and the human ER1. And while they both have regulatory disulfide bonds, the mechanism differs between the two species. In human, the activity is regulated through the disulfide bond that is formed between the so-called outer active site shuttle cysteines and the regulatory cysteines shown here. If they are formed, we have inactive ER1, while when they are not formed in a different way, you have an active enzyme. In Saccharomyces cerevisiae, the regulation is different. You have two long-range disulfide bonds that change the structure or the sterics of the enzyme, but are not involving the active site cysteines. Much less is known about Pichia ER1 and the 10 disulfide bonds that are present within its mature form. In order to elucidate what is happening, Ariana Palmer, who did her PhD in the framework of the Marie Curie ITN secretors, first started by making a sequence alignment. And as you can see here, some things are quite obvious. For example, that these cysteines here are most probably the FFD adjacent interactive site, while these ones here form the so-called outer active site or shuttle cysteines. But for the other cysteines, not much is known. Some of them that are present in human or Saccharomyces do not even have a homolog or a homolog amino acid in Pichia. Some of the other cysteines are conserved in Pichia and in human, but not in cerevisiae. So not really informative from the sequence alignment. In order to find out how ER1 in Pichia is regulated, Ariana adopted an assay known for Saccharomyces, and she mutated all non-active site cysteines to alanine and expressed them in an indicator strain, which is Saccharomyces cerevisiae strain that is especially stress sensitive. And what you can see here is that she performed spotting assays of all these isoforms with a single cysteine mutated to alanine. And when they are growing similar to the wild type or even a bit better, this means that nothing is happening. And these are not the regulatory cysteines, but for two cysteines indicated here in orange, cysteine 76 and the cysteine 332, you see that clearly growth is totally inhibited when you overexpress them, indicating that this is really the regulatory disulfide bond. And we could confirm that there is a single long-range regulatory disulfide in Comagatella, ER1. Also, I can also tell you when you overexpress the same isoforms in Pichia, nothing is really happening, indicating it's just not as stress sensitive or not, it seems to be more tightly regulated maybe than the cerevisial one. The next thing that we have seen is that there is still a cysteine that is not really known. So the next thing we have seen is that Ariana tried to purify ERO and PDI from Pichia. And initially she tried this directly from Pichia, but this was not possible. So she went to another member of the ITN network, Lloyd Rudolph's lab from the University of Oulu in Finland. And she expressed Pichia ERO1 and also PDI in an E. coli strain, especially E. coli strain that can form disulfide bonds. And she purified ERO1 from this E. coli strain. What she could see that actually she could co-purify PDI. And this was a bit of astonishing. And when she performed a reducing SDS page, you could clearly see that this complex was gone, indicating that it was a covalently formed ERO-PDI complex through a disulfide bond. She further set out to find out with which cysteines are responsible for this by mutating, again, several of them to alanines. And she could confirm then that it's the cysteine 136 that is that is responsible for this complex formation. And you can also hear in a structural prediction that this cysteine 136 is clearly in close vicinity of cysteine 166 of the human isoform, which was already shown some years ago to form a complex with human PDI. So as depicted here, we finally set out and also analyzed which of the cysteines in PDI were involved, and Ariana could show that cysteine 136 ERO1 is forming a mixed disulfide with cysteine 44 of the active site of PDI, thereby rendering the complex inactive. So with this, we have allocated all cysteines of Pichia ERO1 with an activity. So we have, again, as for the others, outer active site and an inner active site depicted in yellow. We have one long-range regulatory disulfide bond depicted here in red. And Ariana has also confirmed the presence of a mixed disulfide bond with PDI through cysteine 136, which has not been shown to exist in any yeast before. With this, Ariana also wanted to kinetically analyze the activity of PDI and ERO, and what she could, by using an in vitro refolding assay of BPTI, which is a common model protein for this case, basic plant trypsin inhibitor, as it has 3 disulfide bonds. And this is used as an reduced protein in this study. And then you measure by MS whether you have 0, 1, 2, or 3 disulfide bonds. And as you can see here, even at a low concentration, PDI is a very efficient oxidase, catalyzing the oxidation and de novo disulfide bond formation with a similar activity to the human system. And she could also show that also the isomerization activity of Pichia PDI is quite high, at least much higher than the one shown for Saccharomyces cerevisiae PDI. This brought us to our next question. So PDI is not a single protein. In addition to the canonical, well-known PDI that I've been talking about so far, there are more than 20 PDI family members in humans and plants. And also in Saccharomyces cerevisiae, you have 4 additional family members, one of them being catalytically inactive, but the others can take over the activity of PDI in some part. When we look to the genome of Pichia, we saw that there are also 5 family members. There are homologs to cerevisiae PDI, MPD, and EPS. But in addition, there are 2 more family members that are yet uncharacterized. One thereof was having a ERP38-like domain structure, as shown here. And the second one is a totally unknown PDI family member that has just the domain structure of A and B. And we decided to analyze and characterize these family members as much as possible. So Ariana went back to Olo, and she also again expressed him in E. coli. And what I can say here is that this unknown family member can be expressed, but we have not found what its activity is. And also, I have to say that we have not really seen that it's expressed in Pichia so far, while all the others are expressed to rather high levels. The second one that Ariana looked at was ERP38, or actually what we thought is an ERP38-like protein. It has two thyroredoxine-like domains, an A and an A prime domain, and an ERP29-like C domain that is characteristic for ERP38. However, when we looked at the active sites, in contrast to the known members of this protein class that have two PDI-like active sites, the Comagatella Pathi ERP38 had only one PDI-like active site, and the second one was very glitter-redoxine-like. And this domain architecture is very, very uncommon. It has not been characterized so far. So we decided to rename the protein to ERP41 instead of ERP38 as ER protein with 41 kilodalton. When we looked for homologs in other species, we only found very little of them. One in the yeast field in the Aerobiolitica, a few in Schizosaccharomyces, but not in any other yeasts, a few in some parasites and plants, but nothing functionally related. So Ariana went back to Olu in order to characterize ERP41 in more detail. And the first thing when she expressed it in the E. coli Sidescus strain was that she could see that ERP41 is not forming a complex with ERO1, unlike PDI. However, that ERP41 acts as a catalytic partner for the ERO-PDI complex. And here, ERP1 is even faster than the PDI one, which was already quite fast. So you have a higher catalytic turnover, but a lower activation time for ERP38. Additionally, by dissecting the domains, Ariana could also show that the A prime domain, that is glitter-redoxine-like, has a glutathione concentration-independent peptide oxidation activity, in contrast to the A domain, which has the common glutathione concentration-independent one. So with this, we were wondering what can be the activity of ERP38. And one thing that is known for the human ERO-PDI system is that unfolded proteins bind to the B domain of PDI through the hydrophobic core, thus impairing the interaction with a hydrophobic hairpin on ERO1. And even though this hairpin is not present in the Comagatella or Cerevisiae version, we were wondering if this interaction between the two might be impaired as well and inhibited through the excess of unfolded proteins. So Ariana performed some activity assays again. In this case, a hydrophobic peptide or again BPTI were added as measure for unfolded proteins. And as you can see here, when you add BPTI to the human, the activity drops immediately, and even more so when the hydrophobic peptide was added. In contrast, for the Pichia PDI, nothing is happening when you add either BPTI or the hydrophobic peptide. And while the hydrophobic peptide might just not bind to PDI, we have clearly shown before that BPTI is a substrate for PDI. So we were also wondering, okay, if PDI is not inhibited, maybe ERP41 is. So we also measured its activity in this assay. And again, you can see nothing is really happening when you add the two unfolded substrates. And Ariana could then also show that also ERP41 is able to catalyze disulfide bond formation in BBTI, even though acting as an even faster oxidase than BDI again, but not being able to isomerize it. So with this, we're really wondering what the function of ERP41 is in Pichia. And so Ariana made a knockout, which was very easily possible, indicating that it's a non-essential gene. And most strikingly and a bit unexpectedly, we did not see any growth defects so far in any of the tested conditions. So neither when it's growing on glycerol or methanol, or you have UPR stress or redox stress, really nothing is happening. It's growing like the wild type. So with this, I would like to sum this part of my talk. We have identified or characterized the regulatory mechanisms of Pichia Euro and shown that it differs to the human one and also the known Saccharomyces one. We have one long-range regulatory disulfide, which is compared to at least two in the other two systems. We have an inactive complex with PDI, which is similar to the human Euro PDI couple. However, in contrast to the human one, the Pichia Euro PDI or Pichia ERP41 couple is not inhibited by excess of unfolded substrate. Additionally, Ariana has characterized ERP41 as a novel, very unusual PDI family member with a very uncommon active site combination. She has shown that it's an extremely fast oxidase. Indeed, it's the fastest oxidase that has been reported so far for disulfide bond formation while being a weak isomerase. The activity of its A' domain is independent of glutathione concentration. However, we have not found any biological function yet. With this, we went back to our disulfide bond formation mechanism. And as I've said before, hydrogen peroxide is formed according to all the schemes. And we were wondering, is this actually what is happening during recombinant protein production? So in order to measure it, we need a method that allows to follow the accumulation of ROS, especially hydrogen peroxide, in vivo. And for this, genetically encoded biosensors are an ideal method. They consist of a reporter unit, as shown here, the sensory unit, and for hydrogen peroxide, this a combination called hyper, which consists of a circulated bimutated YFP as a reporter unit, and an oxygen responsive or hydrogen peroxide responsive transcription regulator from bacteria as a sensory unit, that when you add hydrogen peroxide gets oxidized, and with this oxidation, the wavelengths of the reporter unit changes, and you have peak here for the oxidized form in the emission wavelengths that you don't have in the excitation wavelengths that you don't have in the reduced form. Finally, the resource here is a ratio between the oxidized and the reduced signal, meaning that it's a ratiometric sensor that you can also target to the organelle that you like. Another PhD student, Victor Mendez-Morato, set out to use Hyper7 in Pichia, so he expressed it under control of the constitutive promoter in the cytosol, and he could easily show that you have expression of the sensor in the cells, and that the signal for the oxidized form gets stronger when you add hydrogen peroxide. While microscopy is nice, it's not very easy and efficient to measure a lot of cells over time. So Victor decided to use the biolector instead and to record the oxidized and the reduced signal. And what you get finally out of this is this ratio between the oxidized and the reduced signal, which is what I will show you in the next slides. First, Victor set out to add externally added hydrogen peroxide, in this case a very low concentration of 0.1 millimolar, and he could immediately see that you get a signal and that this disappears after a few hours again because hydrogen peroxide is detoxified. If you add more hydrogen peroxide, the signal gets stronger. And if you add even more hydrogen peroxide, up to 20 millimolar, you see here that the signal is not really getting stronger anymore, but it just that it takes longer until it returns, meaning that it takes longer to detoxify this amount of hydrogen peroxide. So after having been very happy that it responds to external hydrogen peroxide, Victor also wanted to measure intercellular hydrogen peroxide formation. And in addition to oxidative protein folding, that we were very interesting, we wanted to validate the sensor with another pathway present in Pichia, which is methylene utilization, which is also reported to be the source of hydrogen peroxide formation during the activity of AOX. So what Victor did is that he recorded first the signal of the wild type cells in glucose conditions, which looks very looks like this here. And when he added hydrogen peroxide, you can immediately see that the signal increases. You have accumulation of hydrogen peroxide, and after some time, it again drops, as shown here. While this is nice, and you can also see maybe the cells also grow a bit like in this case, so Victor decided to look also at different strains of Pichia, having different AOX activities. So in addition to the wild type strain that we have shown also before in red, he also analyzed the mode S strain and also the mode minus strain, which has no AOX activity anymore. And thereby he could clearly show that the activity or the hydrogen peroxide accumulation is due to AOX activity, and that the mode S strains have much less hydrogen peroxide accumulation than the mode plus strain. So after having confirmed that the biosensor works, he decided then to finally look for recombinant protein production. And if you also have hydrogen peroxide accumulation there, the signal here looks a bit different because the growth conditions are different. So this is again the wild type of glucose, and you see a bit of an increase because the cells are always actively growing, very fast growing. You also have more cells here. But what you could also see is that you have a change in the signal, an increase at this point here, showing that the cells were clearly accumulating hydrogen peroxide when they were secreting the recombinant protein, the Fab fragment. Next, Victor wanted to test if he can also get rid of these stressful conditions again for the cells. So he overexpressed some antioxidant genes, and he can also show that compared to the Fab strain itself, the overexpression of the antioxidant genes leads to higher product concentration, so more than 60% more recombinant protein. Nevertheless, when he looked at the cells, he could show that the hydrogen peroxide accumulation got significantly reduced again in these engineered cells, which showed a profile similar to the wild type. And with this, I would like to sum up. So in addition to shedding new light in the regulation of ERO, BDI, and looking for novel BDI family members, we were able to implement a genetically encoded biosensor for hydrogen peroxide formation, to measure hydrogen peroxide formation in vivo in Pichia. And we could show that this biosensor was very valuable to follow hydrogen peroxide accumulation of different strains during methanol metabolism, but also of different recombinant protein producing strains, and that the productivity of the cells get higher when they accumulate less hydrogen peroxide. With this, I would like to thank to all the people who contributed to this work, especially to Victor, who was funded by the Biotop Doctoral School and the Austrian Science Fund, and also to Michael Molin, who hosted Victor in Chalmers in order to set up Hyper 7 for Pichia. Also to Ariana Palma for the first part of this talk, and all the colleagues from the Secretus Network, especially Lloyd Rudolph's lab at the University of Oulu. I would also like to thank the whole lab at BOKU, at the Institute of Microbiology and Microbiotechnology. You might see some familiar faces here. By the way, we have changed our logo. BOKU has changed our logo, so this is our new logo now since a few days. And I would especially like to highlight 3 people here that are working for the Christian Doppler Laboratory for Gross Decoupled Protein Production in yeast and are present at the Pichia conference. So Ben Goldman will present later today, and Karina and Jennifer will present the posters. So I would like to thank you for your attention, and I hope to be live with you in a few minutes to answer your questions.